you very much. I will talk about the role of epithelial barrier in allergic, autoimmune, and other microinflammatory diseases. I would like to thank Beata Rückert, our main technical assistant, the head technical assistant. She has contributed a lot to SIAF, starting from 1998. And it's really great to work with you, Beata. Thank you very much for all your contributions from the beginning of the talk. I also select the laser pointer. I'll first start by introducing type 2 diseases to you, which is one of the main aims for drug discovery and treatment, because they are one of the most predominant and most prevalent diseases of their groups. For example, asthma is the most prevalent disease of childhood. Allergic rhinitis is the main disease for job loss in the world, going up to the prevalences of around 30%. And atopic dermatitis in babies, infantile eczema, is one of the most prevalent disease of babies, which affects around 10% of the babies before COVID times and after COVID times, when babies are in lockdown and without any connection to the environment. The prevalence seems to increase to almost 20%, which is very bad news of COVID, because it will affect a lot of people's lives in the future. The type 2 diseases have several pathophysiological features, which are listed here. And if you learn this list and if you understand the content of it, you will learn more about type 2 disease. So I will go through this list. We have allergen-specific and general type 2 response, peripheral and tissue eosinophilia, goblet cell hyperplasia if there is mucosal tissue involvement, excess and bad quality mucus production, mucociliary dysfunction, remodeling, and at the end, epithelial barrier defect, which in my talk I will mention more about it. So if you look at the 4 or 5 diseases, atopic dermatitis, asthma, chronic rhinosinusitis, mainly with nasal polyps, allergic rhinitis, eosinophilic esophagitis, food allergy, there are similarities in the inflammatory cells and mediators. Th2 cells are quite important, and they have been shown in all diseases, allergen-specific Th2 cells. Type 2 innate lymphoid cells, which were discovered around 10 years ago, are continuously being shown in these diseases. Dendritic cells in the tissues are important, and they are also important, of course, in the lymphatic organs, but dendritic cells migrate to tissues and become part of the inflammation there. IgE-producing B cells and general memory B cell response is taking place in these diseases in the submucosal areas. Eosinophils in the circulation and also on the mucosal surfaces, like allergic rhinitis, you can easily see eosinophils in asthma. You can easily see. In atopic dermatitis, you can see in the circulation, but not in the skin. And basophils are important, mast cells, M2 macrophages, and NKT cells. And if we simply look at the mediators, we have done a lot of RNA sequencing, and in the old times, a lot of gene arrays to look at the mediators, and we can talk about 1,000 mediators of type 2 immune response, but the main mediators are interleukin 4, interleukin 5, and interleukin 13. Interleukin 9 plays a role. Interleukin 31 is important. Interleukin 33, TSLP, interleukin 25, eotaxin, TARC, and MDC, kind of chemokines, and Ig. These are the main mediators. Since you are doing allergy research, you have to know much more than this. But if you are going to give a talk to people, public students, these are the main mediators that you can talk about. And there are drugs and biologicals that we are using currently, and that are targeting, for example, dupilumab, interleukin 4, interleukin 13 receptor alpha chain. And interleukin 5 is targeted by 3 biologicals. TSLP was recently also licensed by FDA, and there is also omalizumab, which is against IgE. So if you look at the factors contributing to the pathogenesis of type 2 inflammatory diseases, we have the immune response, which is the main target and main reason for the pathogenesis. But we have many other things. It is not only immunology, it is many factors that affect type 2 diseases. Epithelial barrier dysfunction is one of the main factors, and there is that leads to abnormal innate immune response, allergen pathogen entry, and continuous damage repair failure. Microbiome and environmental factors are important. They play a role in allergen sensitization. Microbial dysbiosis is part of these diseases, and there is decrease in commensals and increase in opportunistic pathogens, mainly Staphylococcus colonization in atopic dermatitis is going to the level of 90% of the patients, and chronic rhinosinusitis also have 90% of the patients have IgE even development against Staphylococcus. And these Staphylococcus, Moraxella, Pneumococcus are important opportunistic pathogens. And there is genetic disorders that affect the barrier and inflammatory processes. They may influence immune response to different factors, and they are predisposing infection and other effects on barrier homeostasis. And there is defects in epithelial barrier-related genes, mainly filaggrin mutations in atopic dermatitis are the most important and most significant example of this. All of these are playing a role in epithelial barrier hypothesis, which I will talk in the second part of the talk. And we move into a different understanding of these diseases. It's important to know that these diseases are not taking place alone. When you see asthma patients, they have other diseases together. For example, adult asthma has around 30-40% chronic rhinosinusitis. Some 13% have asthma COPD together at the same time, which is also called eosinophilic COPD or type 2 COPD. And they have also allergic rhinitis. And when we see here, allergic rhinitis goes up to 6 to 80% of asthma patients, and ACO is over 30%. And non-steroid anti-inflammatory drug sensitivity is around 7%. So this being common is because, as I have shown you, same cells, same molecules, same receptors are playing a role in the pathogenesis. Peripheral and tissue eosinophilia is an important part of type 2 response, and the drugs which are targeting benralizumab, mepolizumab, resizumab are targeting interleukin-5, and interleukin-5 is the main factor which plays a role in the pathophysiology of eosinophilia. And if you block interleukin-5 or if you block interleukin-5 receptor, then you can block eosinophilia. So there is a release from the bone marrow and mobilization of eosinophils to the blood. This is the first step. And the second step is eosinophil chemokines are playing a role, and VLA4 and YCAM1 are playing a role in the rolling, adhesion, and diapedesis of eosinophils to migrate to asthmatic lung, to migrate to chronic rhinosinusitis with nasal polyps and eosinophilic esophagitis. And they are upregulated by interleukin-4 and interleukin-13. And we have also more on migration to tissues, eotaxin, TARC, MDC, and these are important. And interleukin-5 is important also in the eosinophil survival inside the tissues and together with them, the migration. So drugs which target IL-5 are targeting eosinophilia. They are decreasing eosinophil numbers in the peripheral blood very significantly, and they are also decreasing eosinophil numbers in the affected tissues. But also drugs targeting interleukin-4 and interleukin-13 are decreasing peripheral eosinophilia and also tissue eosinophilia. Another part is goblet cell hyperplasia, excess and bad quality mucus production, and mucociliary dysfunction is part of the type 2 response when mainly mucosal surfaces are affected. We see this in asthma, in rhinitis, and also in chronic rhinosinusitis. Normally, type 2 inflammation and interleukin-13 affect the goblet cell hyperplasia and reduce ciliary function. That's very important to know. And we have also data that we are going to show in the organoids in this area in the same context. And healthy individuals have a normal ciliary bead, and there is a normal ciliary differentiation. But in chronic rhinosinusitis with nasal polyps, there is reduced cilia motility and differentiation, and the goblet cells and mucus production in normal health is taking place. But in chronic rhinosinusitis with nasal polyps and asthma, there is goblet cell hyperplasia and increased mucus production. Another aspect of type 2 response is remodeling. So there is continuous tissue damage and also repairing is taking place because these are chronic diseases starting from the age of 7 and may continue until the age of 70. So the IL-4 and IL-13 are important factors in remodeling. In addition, there's other factors like transforming growth factor beta, activin, bone morphogenic factors. And we have in the remodeling epithelial shedding, goblet cell hyperplasia, mucus production, basement membrane lamina reticularis thickening, together with subepithelial fibrosis, collagen deposition, smooth muscle proliferation, and epithelial barrier opening. They are all taking place at the same time in remodeling. So we have a facet circle that environmental triggers initiate inflammation. Then type 2 cells and mediators are playing a role, and they are migrating to tissues, affected tissues. Epithelial injury and barrier opening is taking place, and together with epithelial repair, airway remodeling is taking place, and also pro-inflammatory mediators are released because of environmental triggers again, and this facet circle continues with relapses, exacerbations of these diseases. And to better understand type 2 response, you have to understand also what is the origin of type 2 response, where was it taking place in the past, in which organisms. And it's important to know expulsion of parasites and larvae. So we go here 90 years back to Zurich University Parasitology Department when William Loeffler was describing the Loeffler's pneumonia against Ascaris, schistosoma, and hookworms. So we follow the numbers in the case of Loeffler's pneumonia. When infective eggs are swallowed, let's say for Ascaris, they reach to small intestine and they hatch. And when the eggs are hatching, one egg ends up with one larva. So the eggs and larvae reach to small intestine, and then they come to liver. And the larva is at the size of 0.5 to 1 millimeter. From liver, they migrate to vena cava inferior, right atrium, right ventricle, pulmonary artery, and they migrate to the lungs. And the larva here in the lungs cause an eosinophilic pneumonia with cough, and very typical eosinophilic pneumonia. It is like asthma, and there is a very strong type 2 immune response, but at the same time opening of the barriers, of the epithelial barriers, and every single larva are expulsed outside the tissue because the larva is small, 0.5 millimeter, one can live with it, but the adult is 10 to 15 centimeters. So when the adults just grow, that will happen within a few weeks, and they grow in the lungs and in the respiratory tract, they will occlude the whole respiratory tract, and this will be a deadly disease. That's why also many animals like the mice, and also that is shown, and in monkeys, it is shown that they have a very strong parasite expulsion response, not to let the adults grow inside the life-threatening organs, but to grow in the intestine. So the larva, when it is expulsed outside the lungs, it is swallowed again. It comes to trachea and it comes to esophagus again, and it is eaten up, and then it goes to intestine, and the worms get adults at the size of 10-15 centimeter in the intestine. There they find the space to grow. Then humans nowadays, it's happening in developing countries, but it was a lot in Switzerland or also in Europe until 1960s. All these parasites were infecting people, and after water sanitation, it showed a very significant decrease, and the parasites are living with humans lifelong in principle, with a little bit of anemia and a little bit of abdominal cramps, and usually with no symptoms. And this was taking place and very significantly. But if the parasites grow inside the lungs, that would be the case, which is a very life-threatening picture, that the adult parasites are coming out of the respiratory tract. Another important parasite which you have to know is Sarcoptes scabi, and Sarcoptes scabi makes the disease scabies, which was very common also in the world. It is still existing, and it makes an eosinophilic dermatitis. So here the sarcop, the agent, tries to invade the skin, and we respond to it with eosinophilia and a type 2 response to just block that the parasite doesn't invade deeper tissues. And with each scratch and eosinophilic dermatitis, the parasite is thrown out of the skin. That's also taking place. So another important factor of type 2 response is you have to know that the prevalence of these diseases were very low before 1960s, but they started to increase a lot afterwards. The environmental influence. So it is not only genetics, but also the environmental influence is the main player of the increase. So we are asking the question, what happened after 1960s to cause the global health crisis? Because before 1960s, these diseases were very low. It is not a matter of diagnosis. It is very important to understand that these diseases were really very low, because we can also look at all the classical textbooks, and the diagnosis of asthma, rhinitis is not so difficult. Atopic dermatitis is not so difficult, but they were not existing too much. And in exposure to epithelial barrier damaging substances, these diseases started to increase after 1960s, and 220,000 new substances have been introduced to human lives with almost no control after 1960s. You have to know that there's a big actually insult to human lives, and anything that we are being exposed in modern lives are new substances to our body. There is no co-evolution, and these are mainly oil products, but also products from other substances, and we are being continuously exposed to them, and we will discuss more on these in the next part of the talk. When John Bostock and Charles Blackley published the first case of hay fever was in 1819, and in 1873 the first 28 cases of hay fever and hay asthma were published by Charles Blackley in 1819. It was difficult for them to collect the cases, and this is true. The prevalence was very low. In 1900, Swiss Army had only 1% asthma cases, and some of them could be also other diseases, but this 1% is true. In 1960s, again, 1 to 2% were having asthma. At that time, we had two major insults, that is the surfactants and enzymes in detergents. And then a second wave came with the entry of dishwasher detergents, emulsifiers to our life. And then the second wave, we started to have food allergy, eosinophilic esophagitis, drug hypersensitivity. They were almost not existing before 1990s. Eosinophilic esophagitis was first described in CF in 1994 and 1996, and the first paper was published at that time. And the first cases were difficult to collect by doctors Uwe Simon and also Alex
Straumann.
Yeah, thank you, William. Alex Straumann, and eosinophilic esophagitis is currently 4% of the population, is affecting 4% of the population. And food allergy was not mentioned too much. It was existing, but it was not a very major disease. It's also around 4 to 5% of the population is affected by food allergy. So the sensitization numbers increased from 1960s to, there are many number of studies, epidemiological studies showing a continuous increase. And the most recent ones are the MEDAL studies, which also CF was involved. There's more than 55% of the population have allergen sensitization. So this ended up with a very high numbers of chronic non-communicable diseases, not only allergy, asthma, but also autoimmune diseases. In 1960s, it is calculated that around 100 million patients were having some kind of chronic non-communicable diseases. And currently, it is known, very clearly known, that around 2 billion patients are having chronic non-communicable diseases. World's population in 1960s was about 4 billion. Currently, it's 8 billion, so it has doubled. But the diseases have increased by 20 times. So that's something that you should keep in mind very clearly. So our work in this area started with first studies on detergents and surfactants and looking at epithelial barriers and epithelial activation. The first studies were done by Mu Xian and Ming Wang and also many collaborators from CF. And we looked at the RNA sequencing years ago, and we found that there is 1 to 50,000 dilution of the detergent, any detergent that you can buy from Migros or Coop. And you can see that there is upregulated lipid metabolism, oxidative stress and cell survival, and downregulated cell adhesion, extracellular matrix organization, and wound healing was taking place in a very high dilution of the detergents, commercial detergents. This is 1 to 50,000. The interpretation of these findings is that a chronic wound is taking place and that tries to survive the cells. So the cells are trying to survive, but in detergent exposure, these are happening. And this was actually published a lot. And the other important factor is emulsifiers that were introduced to our lives. And this is a list of Ismail, a huge list of emulsifiers that one can find in our shelves in Davos and also in USA. And you can see the lists, and they all have E numbers, which are not included in this list. We have to include this also here. And you can see the second page of these emulsifiers, and you can see that many of them are continuously being eaten by us. And Ismail did experiments with polysorbate 20 and polysorbate 80. And these are currently allowed in Europe and in also America. And in America, they are continuously being added to ice creams, baked foods, and milk cream analogs. And they are added in the dose of 1%, which Ismail will show you in his progress report that these are very toxic material in these doses. So what are emulsifiers doing? This is hypothetical, but our research is going on this. Food emulsifiers are thickeners. So they are also sold in the market as thickeners, and they are absorbing the water and thickening the fluid. And the main aim of an emulsifier is when you go and buy tomato juice. In the old times, there was always a water level between the body of the tomato and the water above it, and we were not liking to buy it in the old times. Nowadays, you never see this water level because of emulsifiers added to this tomato juice, and which is prepared in a way that it doesn't, it holds the water in it. So I think that this is the same thing is happening on the mucosal surface of your body, which could be the upper respiratory tract, sinuses, or gut mucosa. So the mucosal lining fluid is becoming thicker. And normally the microbiota, the commensals, should swim in the mucosal lining fluid together with mucus, but they are trapped. And then they don't have a healthy interaction with epithelial cells and dendritic cells. So they are not sending signals to the body for the education of the immune system, and also signals are not coming to the epithelial cells or epithelial cells to microbiome for vitamins and for food digestion. So this is trapped by eating a lot of emulsifiers in our current life, and this healthy relationship doesn't take place, and this ends up with killing of the commensals and decreased biodiversity. So this is taking place with the effect of emulsifiers. There are some studies which are going on from other groups, and we are also working on this, and we will work more and more in this area. And I would like to come here to microplastic and nanoparticles, which are also introduced to our lives. And currently, there's 100 million tons of microplastic plastic produced per year. And microplastic is ingested by cells, which is the work of Julian Virchow with Patrick and Daniel Burla. And it is immune modulatory, it is epithelial barrier opening, and it is binding to receptors. And it is in food, we are eating also a lot of plastic in our normal life. And it was introduced to us as a serious danger for marine species. But nowadays, inner city exposure to plastic and microplastic at our homes from packages is huge. And this we have to deal with, and we have to do research on it. We have to identify the dangers of it. And the work was done with Daniel Burla, Julian Virchow, Patrick Westerman, and Ozan is now collecting together this data and doing the also deep analysis of the RNA sequencings done by Daniel, and we would like to finalize this paper. This is from us collecting the dirt around our house, which has come during the winter, and at the end you can see what is collected. There's a lot of plastic things, and if you can see this big piece of plastic, you can imagine that there's a lot of microplastic also in it, and a lot of other dirt that is damaging and threatening the lives of humans and all the living organisms that we are continuously being exposed. So this pandemic of allergy, asthma, atopic dermatitis, chronic rhinosinusitis, eosinophilic esophagitis is certainly started in 1960s. There is no doubt about it. And the reasons are quite open, and we should certainly discuss and work on it. But in 1970s and 80s, there was almost no IG and IgG response to allergens. So this developed the ignorance hypothesis of Rolf Zinkernagel. And Rolf was saying to us in 1990s when we were meeting in meetings, and at that time we were trying to explain to him that there is a cell subset that's called T regulatory cells are existing. And he was saying, no, we don't need such immune regulation. We have very good barriers, and we are fully ignoring the environment. And he was right at that time. But we were also seeing patients that was related to the barrier damage in the year 2000s, and also IG and IgG response was continuously increasing. Currently, today, allergen-specific IG and IgG response is observed against everything in the environment almost. specific IG is approximately affecting 40% of our population in Switzerland, and more than 90% of these allergic patients have some kind of IG against something in the environment. And IgG, which Rolf was saying there is no immune response, IgG currently is against 100% of the babies at the age of 1 year, even to milk and egg. So at our times when we were babies, we were not showing IgE against milk and egg, But your babies currently, who are recently born and who will be born, are showing an IgG response, an immune response to our main food. Milk and egg is the main food of the mammalians, and we have IgG response to all of them, which is a big change in humanity. You have to understand this, and I don't think there's a bigger change. And we are looking at it from immunological perspective and from molecular point, but this is something which is really affecting the humanity very badly, that we are seeing the effects of it by having 2 billion patients around us. So this is the list of material that we have been interested, and we have worked on their effects on barrier, and we have published many papers on these. And we see here the epithelial cells are not touching to each other, and there is microbes which are going in between or even below the epithelial cells. And here are the factors: laundry and dishwasher detergents, surfactants, diesel exhaust, ozone, nanoparticles, shampoos and body cleaners, enzymes and emulsifiers in processed food, household cleaners like surface cleaners in the shopping malls and at homes, particulate matter, microplastic, toothpaste, and in addition, there's a gluten sensitivity which is increasing. And we have here factors that were existing before our research, and that were humans were being exposed to them, like fungi, allergens, and there's allergens have also enzymes to open epithelial barriers, bacteria, cigarette smoke, mites, and these and viruses. These factors are still continuing, but on these factors, on for example, rhinovirus infection, we have now exposure to shampoos, body cleaners, and dishwasher, laundry detergents, and surfactants, ozone at the same time together with microplastic. And these have synergistic epithelial barrier damaging effects. And this work is going on in the lab nowadays very efficiently. How did we come to epithelial barrier hypothesis? What are the first studies of this barrier hypothesis? In the year 1998, Axel Trautmann joined me as a postdoctoral fellow, and it was my first year as a group leader in CIAF. And with Axel, we started to work on looking at epithelial cells. Axel actually came for eosinophil research to CIAF, but somehow I convinced him to do epithelial cell research. And in 2000, we published the first paper in Journal of Clinical Investigation. It was on epithelial cell keratinocyte death as the main mechanism of eczema. And this was done by T cells, and T cells were opening the skin barriers by killing epithelial cell death. We were thinking at that time, this is everything about the pathology, but now we understand that killing some epithelial cells is a reason of too much inflammation and too much inflammatory cells which are accumulating in the dermis. And this is also reflected in other studies which we did. Sven Klanker did at that time, Thomas Basinski did on sinus epithelial cells, in lung epithelial cells, and together with Axel. And in 2006, we were about to understand the epithelial barrier hypothesis. It is written there, major functions of the epithelial barrier as keep away factors, wash away factors, suppression. And this paper in 2006, the current opinion immunology, is one of the key papers of epithelial barrier hypothesis. Then Mikhail Zoyka and Maciej Szalobinski joined the group, and we started to work on many factors that damage the epithelial tight junctions starting from 2010 and until 2020. And this work is going on. And in the meantime, we started to work on detergents, surfactants, ozone, emulsifiers, particulate matter in disrupting the epithelial barriers. So this is, and we published around 60 papers from my group on this area that are highlighting the major changes. But the first study from Axel Trautmann is quite important to first show how skin epithelial barrier is disrupted by the immune system. Then we came to this epithelial barrier hypothesis paper. This paper is now one of the highest cited papers in very short time. In 8 months, it received more than 70 citations. It will end up with millions of citations, thousands of citations. And I think this will continue because there's a huge interest worldwide. So this epithelial barrier hypothesis explains 2 billion patients with allergic, autoimmune, and neurodegenerative diseases. It shows epidemiological links to exposure to toxic substances. Before this, there was hygiene hypothesis, biodiversity hypothesis, and epithelial barrier hypothesis is not against this. It covers them also. It accepts these hygiene and biodiversity hypothesis, and it explains one step further what is really going on in this epithelial barrier damaged individuals. It demonstrates ways of prevention, suggests future research directions, and needs a huge work from us, from all of you, for public awareness and education. So we have 3 groups of diseases. The first group is the diseases that the barrier on the surface of these affected tissues is damaged. In atopic dermatitis, skin barrier is defective, asthma, chronic rhinosinusitis, allergic rhinitis, eosinophilic esophagitis, inflammatory bowel, and celiac. So the affected tissue barrier is defective, and there is continuous findings of the microbial dysbiosis and barrier defect, and the barrier defect in these diseases is accepted. There is no doubt about it. And the second group is autoimmune and metabolic diseases linked to gut barrier defect. It's not only gut, but it's also sometimes lung barrier defect, and microbial dysbiosis is taking place. There's a lot of publication in these diseases. We have here diabetes, obesity, fatty liver, autoimmune hepatitis, liver cirrhosis, rheumatoid arthritis, multiple sclerosis, systemic lupus, ankylosing spondylitis, and more diseases are coming in this area. And every day we have new publications showing the barrier damage. And in all these diseases, we have microbial dysbiosis in the gut, and bacterial translocation in the gut is taking place. And there's also very nice models that we are continuously discussing and working. For example, gut microbial dysbiosis causes rheumatoid arthritis in mouse models, and there's evidence in humans. And lung also barrier damage causes multiple sclerosis, and there's also very nice evidence in humans. And the third group of diseases is chronic neuropsychiatric conditions linked to gut barrier defect and microbial dysbiosis. The evidence in these diseases is a little bit low. There is direct patient analysis and also post-mortem analysis showing enteric inflammation, barrier defect, and microbial dysbiosis. And we have in this list autism spectrum disorders. These are also very significantly increasing these diseases. They have increased their prevalence after 1980s very significantly. Autism, Parkinson's, Alzheimer's disease, stress-related psychiatric disorders, and chronic depression. And we have microbial dysbiosis, bacterial translocation, and immune response to commensals is observed also even in chronic depression, which are affecting a huge number of humans. So in normal case, we have a homeostatic interaction of the epithelial cells and surface tissues with commensals. There is a live together response, co-evolve response, select and support. I have explained to you what emulsifiers is doing here, or what detergents are doing here. And this homeostatic interaction is broken. What happens now is that these epithelial cells do not touch each other for one reason, which is coming from outside to inside. This is the environment. The second reason from inside to outside, which is the immune system. So microbiome migrates deeper inside the epithelium and gets in contact with basal epithelial cells and stimulates the immune system. So microbiome is not floating on the surface. It is now going in between the epithelial cells, touching the basal epithelial cells, making some changes in the basal epithelial cells, and also passing below and beneath the epithelial cells. And this is called microbial translocation. And this activates the immune system. So this is a very typical picture of asthma, chronic rhinosinusitis. Same is happening in esophagitis. Same is happening in atopic dermatitis, allergic rhinitis. So the immune system is activated, and there is an immune response to mainly microbes that are trying to invade the body. But this immune response is also directed to allergens, pollutants, and environmental factors. And then we are developing these allergic diseases. And this is continuing, and we have the opportunistic pathogens colonizing, and the epithelial barrier is damaged. I talked about staphylococcus colonization, which is very significantly increasing, and translocation. And normally, you should remember now the type 2 response against Ascaris, the expulsion response of Ascaris larva. What we are trying is to expulse all these microbes which are invading our body. Now, these microbes are in the sinus mucosa below the basal epithelial cells. We try to expulse them. What do? We show an eosinophilic inflammation to them, and we try to kill them, and we try to get rid of them, because the immune system doesn't have any eye and doesn't see that these are commensal microbes. If they are below the barriers, that means they are giving a danger signal to our body. So this, at the same time, we have colonization of opportunistic pathogens, Staphylococcus, Moraxella, Haemophilus, Pneumococcus, that are trying to invade the tissue barriers. And at the same time, there is loss of biodiversity. Colonizing opportunistic pathogens and immune response to commensals is taking place. So this summarizes the whole epithelial barrier hypothesis that's taking place in the mucosal surfaces, but same is taking place also in the skin. The last aspect of the barrier hypothesis is that this inflammation doesn't stay in our body as a typical characteristic of immune system cells. They are leaving the tissues and circulating and carrying the message to other tissues. And this is a part of autoimmunity, atopic march, and other diseases. And here we are looking at a section in the lung or a section in the gut, and epithelial barrier is broken. Microbes are between the epithelial cells. These are commensals and opportunistic pathogens at the same time. And they have, some of them have translocated below the epithelial barriers. And there is activation of the immune system. And what happens is that these T helper 17, T helper 1 cells are migrating to brain to cause multiple sclerosis. What happened in 2019 in Stockholm is that there was an explosion of multiple sclerosis cases in the outpatient clinics, and they looked at the severity, and it appeared that in January, all the multiple sclerosis cases became very severe, and there was an exacerbation of the disease. And this was linked to very high particulate matter in the air of Stockholm. And then this particulate matter experiments were done, and it was shown that dendritic cells are activated, B cells are activated, and antigen presentation to T helper 17 cells and migration of T helper 17 cells was shown to the brain of these patients. It's a very detailed study. Another study which was also presented in the year before by Mario Zayas is in the gut, T cells are getting activated and T helper 17 cells are migrating to joints of rheumatoid arthritis patients and exacerbating rheumatoid arthritis. This story is actually a long history, also coming from CF, and Donald Leong worked on this, and it was the work of Mübeccel and her PhD thesis. These CLA positive T cells that are coming from the circulation to the skin that makes skin inflammation. In 1994-95, Mübeccel started to work on this, published the first paper in 1996. And there's evidence for gut barrier defect before diabetes onset, T helper 17 cells migrating to joints from gut in rheumatoid arthritis, and T helper 17 cells migrating to brain from lungs in multiple sclerosis upon particulate matter exposure. So this is kind of a summary slide of epithelial barrier hypothesis. There is genetical and environmental factors and exposure to barrier damaging agents and opening of the epithelial barrier. There's colonization of opportunistic pathogens around this barrier damaged area and translocation of microbiota to subepithelial areas. Then an expulsion response starts, and we are trying to throw away these microbes, and immune response to commensals and opportunistic pathogens is together taking place, and loss of commensals, microbial dysbiosis, and decreased biodiversity is happening, and this disease is becoming a chronic disease, and there's inflammation in periepithelial area and chronicity, and this is followed by defective barrier healing capacity of the epithelium, and chronicity is happening. And this is something which I'm also very proud that this is the first version we developed with CSCM of the skin impedance meter in 2013. Currently, we have this device, which is also the research of atopic dermatitis is going on, and it's for this Nevisense device is for atopic dermatitis, melanoma, and mainly it's licensed in the US for melanoma and skin cancers. There's treatment follow-up is possible for diseases, clinical trial monitoring, and early prediction of treatment response, and early prediction of atopy development. So that's very close to my heart, and it's also the research is going on. I would like to thank very much our co-workers on this epithelial barrier area, and mainly Paulina, Ming, Mikhail, Kazu, Teru, Hide, John, and our collaborators, Roger Lahner, Peter Schmidt, Grander Meyer, Marek Stanek, Claudia Trail-Hoffman, Joanna Gace, Marek Yutel, Liam Mahoney, and Charlotte is now joining this big collaboration group. Nikos Papadopoulos, Vicky, Nina, and Vangelis, Ramazan, and our collaborators from Vancouver, Fumio Takei, Katrin Sher, Itziar Martinez Gonzalez, and from Tokyo, Kenji Matsumoto, and Hide, who was working in SIA for 3.5 years, and Kari from Stanford with her group, Mo Chemji from Imperial College, Lu Zheng from Beijing, and from SIAF, Milana, Arturo, Yasutoka, Anja, Juno, Beate, Lachin, Zeynep, Ismail, Yavuz, Ozan, Duygu, Manru, and Sebastian is joining. And I'd like to thank Mübeccel.